Hey everyone, my name is Renata Franca and I'm a student at UMass Amherst in the College of Natural Sciences Biology Department. I was privileged enough to be able to do research with the Schneider Lab in Pioneer Valley Life Sciences Institute over at Baystate. I hope you enjoy our research. So we were interested in seeing the degree to which aryl hydrocarbon receptor activity may correlate with the degree of transformation of human mammary epithelial cells. We looked at a diverse panel of HMIC lines, 7 of which are normal breast epithelial cells, 2 of which are established immortal lines, and 2 of which are breast tumor lines. So the aryl hydrocarbon receptor, or AHR, was particularly interesting because of its cellular downstream effects. As is shown in the schematic, AHR heterodimerizes with ARNT. Once ARNT, or aryl hydrocarbon receptor nuclear translocation protein, binds to AHR, the AHR-ARNT complex promote the expression of genes in the xenobiotic pathway, such as the endogenous gene family cytochrome P450, or CYP for short. So the CYP gene family play a really important role in enzymatically regulating toxin buildup in healthy cells, as they are largely responsible for the synthesis and degradation of many compounds. Xenobiotic essentially just refers to chemical substances or compounds that are foreign to us, such as drugs, pesticides, and in terms of our work specifically, industrial chemicals and environmental pollutants. Considering the significance ARNT plays in aiding AHR, we were curious to see how different HMIC lines expressed ARNT and if these expressions correlated with the expression of AHR and other downstream genes. So another gene of interest to us was the AHRR, or aryl hydrocarbon receptor repressor. It is believed that in some cells, when AHR overly expresses CYP genes, that this accumulation of CYP can trigger the activation of AHRR in order to suppress AHR activity, which would ultimately repress CYP. Due to its association with AHR and the endogenous gene family, we were interested to analyze AHRR expression in our HMIC lines and see if any correlation to CYP and AHR could be made. Likewise, AHR can also interact with beta-catenin to increase CYP1A1 in the Wnt pathway, but we believe this amplification of genes could potentially contribute to aggressive tumor characteristics. We were curious to see how dioxin-like polychlorinated biphenyl 126, or PCB 126, interact with breast epithelial cells as they transition towards breast cancer. So PCBs are a family of environmental pollutants that used to be found mostly in industrial material, and although not commonly used directly today, its composition makes it extremely difficult to fully eradicate, meaning that it's still found in our environment, especially in rivers and bodies of water. Since PCB 126 bioaccumulates in our ecosystem and PCB is a fat-soluble substance, it affects the food chain. For example, it is advised to eat wild-caught salmon instead of farm-raised salmon because farm-raised have way more fat and can therefore accumulate more PCB. Although we are attempting to regulate the spread of PCB contamination, it is still very much prevalent in the environment and therefore potentially hazardous to us. We hypothesize that there will be a variation between normal epithelium from women in the PCB-induced activated AHR pathway. Also, PCB 126 induced CYP1A1 expression will be higher in tumor cells than normal cells, and that the beta-catenin pathway will be activated by PCB 126 more in tumor cells. We were interested in looking at the response of normal primary mammary epithelial cells. In order to do this, we needed to establish epithelial cell cultures from benign breast tumor, which we were obtained from reduction mammoplasty and mastectomy procedures. We then characterized these cells to determine if they were more basal or luminal-like. With green representing the least expressive and red representing the most expressive, we see which cells express more cytokeratin markers CK5, CK14, and CK18. We also looked at fibroblast marker vimentin. What we see here in figure 1 is that most cell lines were strongly expressive for at least 1 or 2 cytokeratins, suggesting that these cells are in fact epithelial cells. Almost no cells highly express vimentin, suggesting that these cells are not exhibiting fibroblast-like characteristics. We were curious to see how PCB 126 might affect the different cell lines endogenously. We looked at the CYP1A1 and CYP1B1 in stimulated and non-stimulated epithelial cells. And we actually see that for many cell lines, there's a significant increase in CYP1A1 and CYP1B1 mRNA expression in the presence of PCB 126. However, as we will see shortly, the changes in expression of the endogenous genes do not seem to be correlated with increased expression of the genes that regulate CYP transcription. This tells us that although all the cellular machinery needed for transcription to occur is present, which is why we see expression at all, there's a lot of variability among the cell lines, which makes us curious as to what exactly is regulating activity within these cells. We also looked at AHR reporter assay. An XRE luciferase construct was used to see if the reporter activity was compromised or access to promoter sites on the chromatin was different in the cell lines. Interestingly, we did see that some cell lines transfected with XRE luciferase had increased receptor activity in contrast to the non-PCB treated lines, which was lower in activity. Also, the differences among the AHR activity did not always suggest that the variability was in correspondence with the increased expression in CYP1A1 and CYP1B1 in cell lines. This leads us to believe that some cell lines could potentially have a compromised ability to properly sit on chromatin, leading to a decreased expression of AHR and therefore CYP expression, or that other cofactors contribute to the endogenous gene variation. We were also curious to see if differences in expression of cellular machinery were contributing to the variation we saw in the HMYC lines. As can be seen here in figure 4, the expression of these genes were also variable, but appear to be so without really giving us a reason behind the variability. Expression of HR did not seem to correlate with expression of ARM. Likewise, expression of AHR did not seem to correlate with this repressor AHRR. And the differences in machinery did not correlate with the variability in the endogenous genes, which again suggests that there is variation, but not in a direction that tells us why. Lastly, we looked at the beta-catenin target gene AXIN2 across unstimulated and stimulated cell lines. If PCB126 activated beta-catenin, we would expect AXIN2 expression to increase in stimulated cells. No evidence for PCB-induced AXIN2 expression was observed. Furthermore, our hypothesis that the activation of AHR could co-activate the beta-catenin signaling pathway in tumor cells appears to be incorrect. However, our hypothesis that tumor cells would have the highest AHR signaling was supported, with MCF7 having the highest CYP1A1 signaling. Further studies are warranted to understand the significance of variation in the AHR responses.